we're recording this just after Halloween, and is there any horror movie that can match this? I don't think so.
I don't think so, but I think we should probably make one.
Hello, everyone, and welcome back to People, Parasites, and Plagues, a podcast aimed at delivering information about the fascinating pathogens among us from the scientists who study them. I am David Peterson.
And I am Kim Klonowski, your host for today's podcast episode. When it comes to One Health, it's not a one-size-fits-all prescription. It's a holistic approach for a healthy world. Today's guest works to uncover the sometimes surprising factors that influence disease transmission.
Our guest today is Dr. Chris Cleveland, Assistant Professor in the Department of Population Health at UGA and a member of the Southeastern Cooperative Wildlife Disease Study. Dr. Cleveland has worked extensively on both zoonotic and wildlife diseases. Chris, welcome to the podcast.
Thank you very much.
Chris, we wanted to start way back when you were a freshman in college. As I recall, you didn't start out as a science major. What changed?
You were reaching way back, aren't you? And this is being recorded.
We try to do our research.
Yes, well done. Okay, so to Kim's question, spot on. My initial trajectory for my education was actually in the arts. I was a painting and ceramics major and had done that for a number of years, had actually traveled both nationally and sort of internationally to teach ceramics. It came down to a first-year Odyssey course at University of North Carolina, Asheville. That was taught by Dr. Tim Forrest, who's still a good dear friend of mine. He's now retired, but he was an entomologist who looked at bioacoustics, and he changed my trajectory in that first semester through an entomology-focused course that was also very field-intensive. So diving in and getting to geek out on insects was phenomenal. He was also a great advisor. He told me not to take all of my hard courses in my senior year, which I did not listen to. And so I totally had a wonderful undergraduate education. But I certainly learned a lot through that class. And that sort of introduced me into the biological sciences.
Okay, so one of the things you're very dedicated to is wildlife research, and that seems as if that interest started pretty early on. So how did you come to that interest, and how did that bring you to UGA?
Yeah, that's a great question. Through Tim's guidance in my undergraduate career, I had been toying with the idea of pursuing graduate education. And he said, one of the things that you really want to dig into is field research. So going out and being exposed to scientists in the field, graduate students, technicians, wildlife biologists who are hands-on, who are asking questions, spending time with them, boots on the ground, and finding out, well, what are they studying and why? And then digging into that a little deeper, how are they phrasing their questions? Sort of what's that sort of transferable knowledge that they're taking away from the field applications and then maybe bringing into the lab or bringing into theoretical modeling? And so some of my first work for field research started out in Arizona as part of a USGS cooperative unit research project looking at climate change and its impacts on elk herbivory, and then subsequently how that influenced neotropical migratory songbirds. So birds that were coming up from Central South America, stopping over in Arizona. Well, what we were seeing, or what Dr. Tom Martin and his group were seeing, was that elk were beginning to move into higher climbs or higher elevations sooner because snowfall was less over consecutive years. Well, as that happened, they're munching on all of these forbs, all these grasses and everything else. That's also where some of these songbirds are laying nests. So you have this kind of unique impact on habitat structure from a large ungulate in response to climate change that's then really nailing down what's happening at a population level for these songbirds.
So you really have to look at the whole community. You can't just study the elk because changes in their behavior affect other animals in the environment.
Well, exactly right. And yet you still have to study those individuals to assess what's happening on their level. And so do they have a change in their resource use? And does that change then impact health of that singular population? But then what are the resounding effects towards other populations in that ecological sense? So you've got to have two lenses on simultaneously. You have to be able to look at the individual, at the population, and then you have to zoom out and look at the community structure. And so that first field experience really just blew my mind. I went back for a second year afterwards and did more work there. So I had been doing veg counting, which is if you ever want a job that is just going to make you weep in the field, one thing you can do is go out into the woods and look for nickel-sized numbered tags under the leaf litter that are identifying vegetation. And so I'd be out there on my hands and knees, digging through the leaves, looking for little nickels along transects. It was a beautiful test of patience. So I did that some of the time. And then the other time I was small mammal trapping. And that was really my first introduction into hands-on applications with wildlife, going through and seeing what small mammals were around in these areas where elk were eating grasses and moving through, and then how that tied into the bird community. The next year I went back, I was doing more mammal trapping, and then I started to get into more mist netting of birds. I'm looking at eco-immunology and trying to see what the bird populations were.
It's pretty amazing. When you first started talking about elk and then you were previously talking about songbirds, I was like, where are we going with this? But it is a really important lesson, even if you're not doing any type of wildlife type research, right? Even if you're just studying a simple cell, right? You're studying that cell population, but you can't study it in isolation, right? You have to think about what everything else is happening around that single cell. So really important kind of teaching or principle to really nail down early on in your scientific career.
And as an undergraduate, it was my first opportunity to ask a scientific research question. That research question totally bombed. It did not work out well, but I learned a lot from failing. And I was able to actually revisit that and say, okay, well, where did I go wrong in the development of my question? My independent research there was to look at elk herbivory on pollinator populations. So as I had mentioned, I spent a lot of time with Tim at UNC Asheville and was really into entomology and insect populations. And we know that pollinators are in decline. And so I wanted to dig into what's happening with pollinator populations at these higher elevations. Awesome question. I would love to go back and do it again. That first round failed miserably, and that's okay. It was a good experience.
Another great scientific lesson to experience early on and get yourself set for that for happening in the future. Very likely happening in the future, we should say.
Nope.
So we've talked a lot about some of this wildlife and population research, and I'm sure we'll come back to it at some point. But we should probably bring a parasite in, since that's in part of our title. So let's go there. So one area of research you've participated in is part of the campaign to eradicate the guinea worm. Can you give us a little bit of background on this parasite before we kind of dive a little deeper?
Oh, sure. And how much time do you have? Honestly, so this is perhaps one of the best known parasites. Definitely not from a wildlife angle. And so when you're speaking to someone whose training has been staunched more in a wildlife arena, to come at it from a public health menace or a public health scourge, or some of the terms that have been used to describe guinea worm or Dracunculus metanensis, it has a pretty fascinating historical tale that goes back for centuries and centuries. So dracunculiasis, or infection with guinea worm, or Dracunculus metanensis, has been documented in ancient Persian medicinal texts. There are lithographs and prints that show that sort of stereotypic or very classical removal of the worm from an afflicted leg or appendage of a human by winding that worm out on the stick. And I think it's important to mention now, too, if we're talking about this parasite, it's a nematode, so it's a roundworm. The females are the only parasite that actually emerged from the infected host, and we're going to stick with humans for now. That female, when she comes out, or when she's finally removed, is 1 meter long. So it's a very large parasite.
So our listeners, think about that for just a minute. 1 meter long worm in your leg. There's just an inch or so sticking out at a time. When it senses water and wants to lay, it lays. The eggs are larvae out in the water.
These are free-living first-stage larvae, and they are expelled through sort of an opening. The female, the immunological response in the body doesn't sort of show up until the very last stages of gestation, just prior to emergence. And what that female does is she shoots a few larvae at the skin, which then begins to shed back the dermal layers until there's something that looks like a little volcano. And then she averts her uterus through that and comes out when she senses, just as you said very deftly, water. So there is a change in temperature that this parasite senses. And when she knows she's in water, then she shoots out about 500,000 free-swimming first-stage larvae. Like, it was Mueller back in the 70s who actually sat and counted out all of these free-swimming babies.
So we have a larvae gun that's being shot at the skin.
It was like a larvae spaghetti gun. Yeah.
So we just...
Words I never thought I would ever hear.
We're recording this just after Halloween, and is there any horror movie that can match this? I don't think so.
I don't think so, but I think we should probably make one. But I think too, do you need a horror movie? Right? Like this is real life.
Yeah.
And I think one of the things, so as I mentioned, we're talking about the history of this parasite. It has been documented in the Old Testament, right? So I think there's reference to the fiery serpent and Moses and people trying to encounter and deal with that. This parasite has been around for a very long time and has been predominantly associated with human infection. It was in the 1980s when President Jimmy Carter, the World Health Organization, and the Centers for Disease Control and Prevention started to get involved with eradication and management of this parasite. And so at that time, there were 3.5 million cases. And these are all cases in humans. And I'm drawing a blank about the number of countries, but we're looking across sort of the sub-Saharan belt of Africa over into India, so covering the Middle East. So maybe 26 countries at that time that were endemic for guinea worm transmission. I think it's important for everyone to know this isn't a parasite that will kill you. You ingest the third stage larvae through drinking water that contains the obligate intermediate host, which is a copepod, so a little water flea. And these water fleas are ubiquitous. They're found the world over. There's a very specific group called the cyclopoid copepods. They suck up those, we call it a larvae gun. They suck up that cloud of first stage larvae and they eat them like spaghetti. Those larvae bust out of the gut of the copepod and they hang out in this intercarapace space for about 2 weeks where they molt to the infectious stage, the L3. Then you or I or someone in a village will go down to the water and get a bucket, get some receptacle filled with water, and you'll drink it. And you have no idea that you just ingested infectious larvae. It takes 10 to 12 months for those larvae to mate, to develop into that large gravid female. So she's pregnant, she's got her babies inside, and then to begin to emerge. So 10 to 12 months where you really don't know you're infected. There's no
symptoms.
No symptoms, no immunological response. There's no swelling. You don't really have pain until that female is as large as she's going to get. And she's really started to migrate through. Now, I'm not speaking from experience, right? So I can't say exactly what that feels like. Mm-hmm. It's called the fiery serpent for a reason, right? So when that female is as large as she is and she's migrating through your leg, below the subcutaneous sort of fat, on top of the fascial linings of your muscle, you are starting to feel like your leg is on fire. And what's, I think, very fascinating about this parasite in the human is that that's a parasite-mediated response, driving the infected host to try and submerge that afflicted appendage in the water. Going back to David's point where she senses thermal gradient change, she feels water, and then she can shoot her larvae back out, and that cycle continues. So in the 80s, we're talking about 3.5 million people who are afflicted with that. As I said, it won't kill you. But when you are infected and when you are dealing with that sensation, it's really hard to work. And the predominant countries that were experiencing this parasite are low to middle income countries, very agrarian based, right? Needing to get out and work their fields and crops, and needing to be able to go to market to sell things like coal or fish, needing to be able to put food on the table and care for their families. Well, you can't do that as well if you're infected and your leg feels like it's burning. The maybe more risky consequence of infection is the secondary bacterial infections that can happen. So now you have an open lesion or an open sore in an area that's really remote. These are places that don't necessarily have readily available access to medical care, may not have sterile bandages around, may not have that sort of intervention, especially in the 1980s. This wasn't necessarily on everyone's radar. So being able to provision that to people was really hard. So it's the secondary bacterial infections, the fact that infection with the worm can turn into something much greater where you're looking at loss of limb, which can be extremely dire.
So since the 80s, and I guess I'm jumping forward and giving away the punchline a bit, but the folks with the guinea worm eradication program, they've brought the number of cases down to a literal handful. I think last year there were 13, 14.
Right, and that in and of itself is a phenomenal success story. I think. Oh, go ahead.
I was just going to ask, can you tell us how they've done that?
Yeah, and the way they've done that is phenomenal. We can think about what's been eradicated thus far. So smallpox. Mm-hmm. And do you know the other one?
Rinderpest.
All right. Okay, so what do those two have in common?
Single host.
And what else? What type of pathogen are they?
Viruses.
Viruses.
Yeah.
So we can look at ring vaccination trials.
Mhm.
We can surround infected hosts, individuals, territories with a vaccine. There's no vaccine for guinea worm. In terms of one of the biggest challenges facing the guinea worm eradication program right now, it's having suitable interventions and diagnostic assays. And we'll kind of get into why that's become so important. For humans, the most effective interventions to interrupt transmission with guinea worm were low cost, basic, and deployable really easily. And so I think we have to harken back to some of the forerunners in guinea worm eradication. That would be Dr. Donald Hopkins, who was involved with smallpox eradication, Dr. Bill Foege, Dr. Ernesto Ruiz-Tiben, and then Dr. Mark Eberhard, who was formerly at the CDC. He was actually my mentor through my dissertation and a phenomenal parasitologist. Those interventions that they came up with, education. So get out in the villages and just start talking to people. Show people that this is how transmission happens and how we can interrupt it. That still continues till today. Following on the heels of that would be provisioning of clean water. So there's three different methods that kind of follows suit. One is putting in a borehole well in some of these villages. And so some of these areas have no access to water. If we look back historically to the cases that were happening in India, India had step wells, and some of those step wells would be contaminated. So educating people about, hey, this step well has guinea worm larvae in it. It needs to get treated. We'll come back to that. But so the education and provisioning of clean water, and then water filtration. And so low cost, easy water filtration takes sort of two approaches. One is a cloth filter that can be put over a water jug. And with a pail, you can go and scoop the water out of the water body, run it through the filter. That filter is the correct size to eliminate or remove copepods from the water. So you know that the water that's in your water container doesn't have copepods in it. The second is a life straw. And there's a company that has graciously volunteered to donate hundreds of thousands of life straws to people in endemic countries. They're light, they're portable, they're worn around the neck, and inside that life straw is a very simple screen filter that keeps copepods from becoming ingested. The other intervention that's been really successful is the use of Avate or Temifos. So this was a larvicide originally developed for targeting larval mosquitoes and fighting malaria. This also happens to be really successful at nailing copepods, that intermediate host. And so if you can decrease copepod populations in an area that has a high rate of guinea worm transmission, then you can sort of quell down transmission to the human hosts. And then the final sort of feather in the hat of all of that is active case containment reward system for identifying where this is happening, and then community involvement. And so a lot that happens is if someone is infected, they can self-report, they can go to a containment center, they can receive a per diem. There's usually a reward of food or soap or a cash reward for self-reporting and acknowledging that was an issue. And you're effectively taking the infected individual out of the system, and they are not transmitting more parasites or leading to subsequent infections in that areas. So you're right. 1980s to now, we're looking at a 99% reduction of human cases, and that is outstanding. And so I think a lot has to go to the credit, the energy put in by the Carter Center's Guinea Worm Eradication Program. They're partners with WHO, they're partners with CDC, but we really need to honor and appreciate the amount of effort put in from the ministries of health in all of these countries. None of this would be possible if you didn't have country buy-in. And so if we look at Ethiopia, we look at Chad, South Sudan, Mali, Angola, just listing off some of the remaining endemic countries, each one has a program. Each one has people who live in country, who are native to country, who are in those villages, who are out trying to spread the word, who are the most critical, fundamental part of guinea worm eradication. And yet they're not the ones who are always noted as being the ones who are driving eradication.
So when I talk to a class about guinea worm, I make that point about how important the folks in the country are, because I said, here's an example. Let's suppose someone outside of the U.S. knocks on your door and starts telling you what to do, versus having your neighbor come over and say, hey, here's some information that'll keep us all safer and healthier. Mm-hmm. So
it's really important. And I think that's another point just to touch on, as someone who's had this opportunity to go internationally and work in areas afflicted with guinea worm. Most of my work happened in Chad, Africa. I do not speak great French. My Arabic is limited to maybe 2 or 3 phrases. I come in as an outsider. And so I think as scientists, appreciating that we're on the heels of a colonialism era of science, coming in thinking we know best, thinking that we're going to come into a village and tell you what to do and what's going to happen and how this is all going to work out. We're not even going that far, not even communicating with the villagers. I think a lot of what the Carter Center does extremely well is prioritizing that. And it was an amazing experience to go in and sit in a village center that was so far removed from anything, to sit with a village chief, to sit with the elders, to sit with the community, and through multiple tiers of translation, explain who I am, explain what I'm doing. That's one side of the coin, though. The other side of the coin is to sit there and hear the questions from the villagers, to take the time and to be patient enough in your research agenda to prioritize that and say, what are your questions? How can I address them? So that there's community level buy-in.
And I think that's really important in thinking about, so one of our previous guests, Glenn Nowak, talked about listening. And then there's this context of being an active listener, right? So listening and hearing what people have to say. And I think sometimes as scientists, we feel like we have to be constantly teaching or telling, right? And not necessarily listening. So that's another, of course, great skill to have, right? To get the science out and to get, something as important as how to treat and prevent this infection from happening, to kind of take hold in those types of situations.
So, Chris, we talked about how the number of cases have dropped dramatically, 99% plus, which is a tremendous success. But there have been a few sticking points and a few surprises that this parasite has thrown at us. Maybe you can address those.
Yeah, certainly. Yeah, nature is full of surprises. And so to that end, one of the things that happened was in 2012 in Chad, after a decade of there not being reports of guinea worm in humans, a human reported. And then on the heels of that, a domestic dog was found to be infected. And so now we're ringing alarm bells. Now we got red flags left and right. What's peculiar about this is that this is highlighting maybe a one-off. So maybe this was an aberrant transmission event. What began to happen to 2013, 14, 15 was exponential growth in the number of dog infections that were being found and reported in Chad, Africa. A lot of questions that come when I'm presenting on this or when we're talking about this as well, is the number or the increase in cases driven by the fact that you found that first dog and now more people are looking? Potentially, yeah. That's something that has to be acknowledged, and we're going to account for some degree of increase as a result of increased surveillance. However, over the history of guinea worm, there had not been reported this number of dogs infected in any country, nor had there been infections in cats or baboons that had also been reported. So we kind of move forward through our timeline. 2012 in Chad, after this 10-year period or so of no cases being reported, a human shows up. Then a dog is found. 2013, 14, we're moving into 500 to 1,000 to 1,500 cases or infections being reported in dogs. And yet human cases are still really low. And the human cases are not showing up in the same areas that dog infections are showing up. So now Dr. Mark Eberhard published this great paper called The Peculiar Epidemiology of Guinea Worm in Dogs in Chad. It's peculiar because those two scenarios where humans are getting infected and dogs are getting infected are not co-occurring spatially, meaning those infections are coming from different areas in different water bodies. And it's appearing, right, that dogs are maintaining that transmission among themselves. So then 2014, we've got baboons that show up infected in Ethiopia. They're also in areas where baboons are considered sort of a pest. They're coming in, they're destroying ag crops, but some baboons are being found with guinea worm. And then we're having domestic cats show up on the scene in both Chad and Ethiopia. So now you've got a host or a parasite that was really thought classically, historically, throughout all the textbooks to be really limited to humans. Within the span of 4 to 5 years, we blow that out of the water, and all of a sudden we've got 4 or 5 hosts at play in these endemic countries. That's a really big problem. It's a really big problem because some of the initial questions are, well, are they the same parasites? How do we know that the guinea worm in the dog that's showing up that's similar, it looks like human, is in fact this same parasite? Well, the book has been sealed on that. So Dr. Liz Thiele, who's at Vassar College, has been running all of the genomic work on these parasites. And she can get down to a degree of detail that will show sibling relationships between parasites. And that's really important when we look at the degree of diversity and the amount of transmission that's happening. So if we have a high degree of genetic variability among these parasites, it means that there's more transmission happening. And she can show, and she has shown, along with colleagues at Wellcome Sanger Trust, that without a doubt, irrefutably, the parasites that are showing up in baboons, that are showing up in cats, that are showing up in dogs, are the same parasite that were showing up in humans. It's all guinea worm. From an eradication standpoint, what does this mean? And that's where the fire has been lit under our rears, so to speak. How do we approach this situation? How do we start to interrupt transmission in dogs, in cats, in baboons? Because human cases are still low. The guinea worm program and its partners are really effectively managing human cases. That's phenomenal. It needs to continue that way. Getting our hands on dogs, getting our hands on cats and baboons, that's been the much more significant challenge. We can talk some about the current interventions that are being put in place to do that. Happy to dive into that.
Sure. I mean, we're interested in learning about how you kind of approach this One Health, right? So the surveillance in the field, like how is that being done?
Right. Remember, I asked you how much time you have.
Exactly.
So I can keep going until this podcast is now 3 hours. Listeners, I'm sorry. So I think some of those initial issues are, let's identify whether or not this is the same parasite. And then what are we going to do about it? If you recall, I said there's no vaccine. There's no shot to take. Right? So we are really focusing in on what are going to be the elementary steps of transmission. We still didn't know at the time, and I would argue that we still do not know now, how transmission is occurring to dogs, cats, and other animals in Chad or in Ethiopia. So some of the initial work where I became involved was starting my PhD on this work. So that was in 2016, and began working with Dr. Michael Yavsley at the Southeastern Cooperative Wildlife Disease Study and Dr. Mark Eberhard. And in his paper that I referenced earlier, The Peculiar Epi of Guinea Worm, Mark brought up this question. It's like, what if there's something else in the water that could be moving infectious larvae along different trophic levels? And so what if there's a fish or a frog, a tadpole or a small fish that would predate copepods containing infectious larvae that would then be consumed by a dog or a cat or a human? Some of my dissertation work really focused in on that. And we were able to show in the field and in the lab that frogs can become infected with guinea worm larvae. And when you feed that to a model host, those hosts can also become infected. We were able to show that fish can ingest copepods containing larvae. You feed that to a definitive host or the host that's going to lead to patent infection, those hosts can become infected. In the field, we were able to find guinea worm larvae, those third stage larvae in frogs. And so if you're talking about a needle in a haystack, this is like taking that needle and chopping it into 1,000 bits and throwing it out there. So we were really lucky. What didn't line up for us was the amount of infectious larvae or the number of infectious larvae we were finding in frogs. To correspond that to the dogs would have had to eat this unrealistic number of frog protein, and they just weren't. We also kind of asked the question, and a lot of people were bringing this up as well, why can't dogs just get infected the classical transmission route? Why can't dogs, cats, just become infected through drinking water? And so we've looked at that a number of different ways. And this gets into the fluid dynamics of dogs drinking water. So if you haven't looked up videos about how a dog drinks water, it's really fun. I would say go and do that. Right? But a dog doesn't have opposable thumbs. They can't go down and just scoop up a bunch of water, and they don't use straws. What they do is they punch their tongue through the surface of the water and almost in a reverse action, throw water back into their mouth. That's really important. That disturbance, that ripple effect, causes sort of a fleeing behavior in copepods to move away from the source of disturbance. So if a dog walks into water and they're drinking from the top, few centimeters of water, they're going to be punching that surface. They're going to be causing a ripple effect and, in essence, driving copepods away from that area. So we tested that here at UGA and found that dogs could, in fact, ingest copepods. And mind you, these copepods were not infected. We were just trying to see whether dogs will ingest copepods through a normal drinking event. They can. But you have to spike those copepod levels so high in the water body that you do not see that in the natural environment. So we were able to make something happen in the lab where dogs can ingest copepods, but it doesn't hold up in real life. So we don't necessarily think that dogs are just out in the wild in Chad drinking water and acquiring infection that way.
That's really interesting because I don't think I would have thought to investigate it as far as you did. I would have thought, okay, there's infected copepods in the water source. Humans are getting it there. Obviously, the dogs are
just drinking. Mm-hmm.
Right. So,
it all ties back to humans are not getting infected at the same location where dogs are. So what is the challenge here? How are we really trying to nail transmission to the wall for these animals? I think an important thing to mention here, so that's the classical route of transmission. Then we're going to look at these other aquatic animal hosts that could be moving infection. Chadians, and I'm going to stick with Chad because it's the country I know best. It's the place where I've worked the most. I'm not going to speak to some of these other countries, but Chadians rely very heavily on fish protein. And they have fished extensively through the Shari River, which is the predominant water body moving through there, and their oxbows and other lakes that have fish populations. And they will seine everything out, big fish and small fish. So as I mentioned, those small fish can hold larvae, can be infectious, and people will eat small fish just as much as they'll eat large fish.
Mm-hmm.
Cats will also eat small fish just as much as they'll eat large fish. Dogs will eat small fish just as much as they will eat large fish. That was a question we had during my doctoral work in Chad, and we were not able to find guinea worm larvae in the entrails of fish. We were at markets, we were collecting fish from ponds, we were looking high, low, thither and yon for that small piece of the needle in the haystack, and we weren't able to find it. So I think that research question still stands and harkens back to the larger issue here. From a basic biological or parasitological perspective, we still don't know how transmission is occurring to some of these animal hosts. And that's fundamental and critical when we look at an eradication campaign. One of the biggest challenges, and this is acknowledged, everyone in the program knows this, is every intervention that's thrown out in a village, whether it is going to be water filtration, whether it's going to be abate, whether it's going to be burning and burying fish entrails or frog entrails, because that could be a risk. If we're going in from a scientific perspective to study those villages, not a single one of those interventions is tailed back or removed. We cannot risk having more transmission happen. And so we play full court press all the time. From a scientific perspective, that's a challenge because we've got these overlapping covariates. And one could be more significant or one could have a more profound effect than the other, but it's really hard to tease it out. At the end of the day, we don't want anyone to get infected. And so we're not going to let off the gas, which is why it's really important to have these coupled laboratory studies and sort of evaluate what's going on in a lab and see what holds up.
It must be a challenge also to keep the momentum going at all the levels. So I could imagine, say, in the Chad Department of Health, they have a lot of things on their plate. They've got malaria, they've got other diseases, they've got all the standard childhood diseases, they've got limited resources. And the argument might be made, well, we're mostly taking care of guinea worms, so let's divert some resources. And yet, as you said, you need the full court press.
Mm-hmm. And I think the benefit here, and that's a fact that has been brought up, and it's a good one. If we're managing guinea worm infections in humans, do we still need to pursue eradication? And how much do we need to put into fiscally or energetically into the animal infections? The benefits to continuing the eradication program are that overall positive impact on health and well-being in these communities. And it isn't, as you just mentioned, malaria, other neglected tropical diseases, polio, the ability to have infrastructure and energy at the country level to address and be involved in these communities is phenomenal. And so you don't let up yet until you've achieved that goal. And we're not giving up. The Carter Center certainly isn't giving up. And there's not a country out there that's ready to throw their hat in and be like, no, we're done. Because it does speak to a larger issue of how do we address critical health issues in some of these communities that might not otherwise be getting served. So how do we lead to a more healthy society in some of these places? And the Guinea Worm Program does a pretty phenomenal job addressing that. Their target might be eradication, but you go into some of these villages, it shows energy, it shows effort, and it also shows interest to the country and the country's health programs that there's people out there who care. Having that holistic approach speaks to, I think, what Kim brought up earlier with a One Health system. We're no longer thinking about a single entity, a single pathogen, a single parasite. We do have to think a little bit more broadly and holistically when we come up with solutions or when we design and ask our questions and mobilize together in the field, being broad and looking at the greater positive outcomes becomes really critical and helps us, I think, just fill some important gaps from a scientific perspective.
I think I want to end that note by asking a fairly simple question. So it seems like you've done a lot of multifaceted studies, really addressing, we've talked mostly about the guinea worm problem today, but you've had the opportunity to work with some really interesting people to address some of these scientific questions. And I think a lot of our listeners, particularly graduate students who are just sitting in a lab, working on something, don't think about maybe this cooperation you have to get something really done out there in the world. So I want to know, how has your experience really affected the way you do science or the way that you teach science to graduate students?
I think that's a great question. And to any graduate student out there sitting with a sore thumb from pipetting or banging their head against the laptop as they're trying to run their R code and all they're getting is red errors, do know that science doesn't happen in a silo. Science is inherently interdisciplinary, but you have to be willing to say, I don't know this as well. I can go and ask somebody else. And more often than not, that somebody else is really going to be excited and wanting to help. And what we find from creating these interdisciplinary, transdisciplinary, multidisciplinary, collaborative approaches is that everyone has a different perspective they bring to the table that can help you ask the question a little bit different, that might change the interpretation of your results, that might lead to the ideal outcome or a surprise outcome. And so through my work, I was able to work with public health epidemiologists. I was able to work with other parasitologists. I was able to work with pharmacists, pharmacologists here at UGA. We still collaborate broadly with folks at Texas A&M University, Ohio State University, Vassar College, Gates Foundation. There's no shortage of people who come to the table. Virologists now have a stake in the game. And so through the guinea worm work, I found that there's a lot of people who have extremely valuable perspectives and experiences that if they're not as familiar with the system, that's almost more prized because they can come in and take the blinders off that you might have. I may have been working in the system since 2016. I was just at a meeting on Monday and Tuesday with the Carter Center where they brought in folks who were engaged in polio eradication, the polio campaign, and they asked some phenomenal questions. Like, oh yeah, we hadn't thought about that. Most importantly, as a growing student, I think it's being able to be open and accepting of ideas that are counter to your own, and opinions and ideas and approaches that are going to be counter to what you think is right, because everyone can be right. But if you don't give space for those other ideas to grow and breathe, you might not be advancing the best possible way. So having that diversity of team members, not just in science, having that diversity of team members from various cultures, I think this harkens back to what I was saying earlier about really engaging at the community level. Some of the coolest lessons I learned were in field working with people who are involved with cattle health in Chad, and just talking about cattle movement and how that impacts dog movement, how that impacts human movement. So really being broad in the questioning and being able to talk to people who have so much different types of knowledge than I did or ever would think would be relevant. Turns out, hey, they made a great point. We should ask that.
Well, I think we're going to end there today. Our guest today is Dr. Cleveland from the Department of Population Health and the Southeastern Cooperative Wildlife Disease Study. Chris, thanks for being with us.
Thank you. It was my pleasure.
Links to additional information about Dr. Cleveland's research can be found on our Instagram account at PPP Podcast UGA.
Thank you for tuning in today. Please rate the podcast, and if you have any questions or suggestions for future episodes, reach out to us via email at ppp@uga.edu. This podcast is brought to you by the Faculty of Infectious Diseases and the Grady College of Journalism at the University of Georgia. It is supported by the University of Georgia through the Office of Research, the UGA Graduate School, and the College of Veterinary Medicine. Thanks to the New Media Institute at Grady for the use of their Studio Not Found podcast facilities, and special thanks to our production assistant, Sid Wigand, for research, editing, scheduling, and in general just keeping us on track.